I'm Catriona Jamieson. I go by Cat Jamieson. We are thrilled to be able to work together, thanks to Danny Sanford and his tremendous vision and generosity over the last many years, actually since 2013. But back in 2002, we started the Sanford Stem Cell Institute to really accelerate the ability to make new therapies from the tremendous scientific discoveries that are made here at UC San Diego and beyond. The reason to have the Medicine Informing Novel Discoveries Mind events is to really change how we talk about science and medicine and make it more palatable for a broader audience, but specifically for patients. We're all going to be patients someday. So you really hope that we can make new therapies and that they enhance endogenous repair and regeneration. So what I mean is take our own stem cells that are part of our bodies already and make them work better for us. And if they're not working well, then replace them, fix them. So we'll have two great talks today. And then also we'll have Tiffani Manolis on the panel, who's here from Thermo Fisher. Thermo, as you know, makes all of the reagents for science, all the stuff we need in our cookbook for science, so that we're really glad that you're here, Tiffani. And then we'll have a patient story to take it back to why it matters. And this is essentially why we're so thrilled to work with the Museum of Contemporary Art here in San Diego to really showcase how we bring together the art of science and medicine. So for today's Mind series, we're talking about stem cells and the next generation of regenerative medicine therapies. Now, I know we all have different backgrounds. How many people here are scientists? Quite a lot, actually. How many people are physicians? How many people are in business? Hey, all the same people raise their hands. Wait a minute. We live in a unique community. How many people are involved in the administration of clinical trials? And how many people are ultramarathoners? Had to get Matthias to raise his hand. Anyway, we have an incredibly energetic, talented team here. We have people who come here for just about every Mind event. So Lee and Juniper are here. Great to see you both. My mom is here, and she's going to ask very targeted questions to everyone that's here. Hello. And, of course, Sheldon representing the Sanford Stem Cell Clinical Center. I think you're going to get an action-packed view of what we're doing to distill the discoveries that happen at the bench side and how we transition them to the clinic with alacrity. So first of all, we're going to have Dan Kaufman speak about all the advances that he has made on the cellular therapy front in a very clever way, and what he's doing more recently with different vectors to get the payloads into cells more effectively. Dan Kaufman has been a leader in the pluripotent stem cell space. He worked with James Thompson, who was the first to make human embryonic stem cells, and Dan Kaufman was the first to make hematopoietic stem cells out of those human embryonic stem cells. We were thrilled to recruit him here from the University of Minnesota a few years ago. And since coming here, he started the Advanced Therapy Lab and now has grown it together with Christman, our second speaker, into a whole center within the Sanford Stem Cell Institute, so we can accelerate not just cellular therapies, but extracellular matrices and different products to really get stem cells to work better. So we're going to hear from Dan. He has the endowed chair now, thanks to Danny Sanford, but also his own talent and productivity in making the first induced pluripotent stem cell-derived natural killer cell product to ever be used to target cancer. Ever. So that's Dan's discovery. We just want more of that. Yes, please, Dan. So he's going to tell us about the new advances that he's making with the Advanced Therapy Center and also his lab at UC San Diego. So Dan Kaufman, please join us.
Thanks for the really nice introduction. Thanks for everyone to come out and hear about what we're doing. What I'm going to try and do in the brief time that I've been allowed is to give I know it's a very diverse audience, give a really brief overview of cell and gene therapy and where the field is at in general, and then talk a little bit about my own work, which Kat introduced, and then the new initiatives with the Sanford Stem Cell Institute on how we move more of these cell and gene therapies into clinical trials here at UCSD and potentially other places. So cell and gene therapy, as I have here, is really the idea that's rapidly advancing to replace or repair diseased or damaged cells with healthy cells or genes. So there are, if you can actually go to the FDA website, there are about 45 approved cell and gene therapies by the FDA, probably a similar amount in Europe as well. And so these can be things like gene therapy, where they actually inject vectors, like typically actually engineered viruses, that can replace genes that are lost. So in something like hemophilia, which you've probably heard of, which is a bleeding disorder due to a loss of a clotting factor, you can actually inject the vector into the liver. The patients will make this clotting factor, and they don't need to have the factor replacement. Retinal disease are genetic retinal diseases. They can actually inject these into the eye. There's others for spinal muscle disease and so on. My area is in cancer therapy, so I'll tell you briefly that there's a number of now cell therapies for cancer treatments, and then a lot of clinical trials for making dopaminergic neurons for Parkinson's disease, pancreatic islet cells for diabetes, and so on. And in some cases, these are combined. So there's now an FDA-approved therapy for sickle cell disease, where you can actually take the patient's blood stem cells, correct the sickle cell mutation, give them back to the patients, and they're essentially free of the disease. It's really remarkable. Similar process for immunodeficiency. So this is the bubble boy disease, if you all hear of, and you can essentially cure kids from those type of things. And so this field has been rapidly growing. This shows the clinical trials over the last 10 to 15 years. The graph on the left is actually the cell therapies. The graph on the right is gene therapies. And as you can see, there are now about 10,000 clinical trials worldwide per year in these type of therapies. The other thing that's interesting, besides just the overall growth here, is if you look at the little key, so the blue lines, the vast majority of these trials are here in the US. The gray line is China. Okay, so if you look 10 years ago, 2014, 2015, hardly anything there. China is just growing astronomically as well. And what's also sort of equally surprising, Europe and the UK is in the top part in the blue. Not so much there. So there's not as much innovation as there is here or in China, which is really where the majority of this work is going. But again, these trials continue to escalate and grow. So again, for my field of cancer cell therapy, I also do clinical work in hematology, cell therapy, bone marrow transplant. So how many of you have heard of CAR T cells, right? So this is a big area. So most people have heard of this by now. This is, over the last decade, has really been one of the biggest advances in this field, where for certain types of cancer, and this is one of the key points, it's really limited to certain so-called B-cell malignancies, so types of leukemia, lymphoma, as well as multiple myeloma. This treatment can work very well. And what it involves, I'm not going to go into detail, but it's a patient-specific therapy. So you have to collect cells from the patient, engineer them in a special lab like we have here for our GMP facility, which I'll tell you about, grow these up, give them back to the patients. Takes a few weeks, and it costs $500,000 per patient just to make those cells, right? But it can be very effective. About half of patients with otherwise incurable cancers are actually cured from this type of disease. So it's really been great, but there's challenges, right? So it only works for certain types of cancer. It's complicated. It's made on a patient-specific basis. It's toxic. And while we like taking care of these patients here at UCSD, if you live in somewhere like, Bakersfield or El Centro, it's hard to get here, right? So you have to come to specialized centers. So really part of the work in my lab has been to make this type of cell therapy easier and to make cells as easy to use as a drug. I like to think about it that way. That these can be targeted so that you are killing specific types of cancer, but they're standardized and quote unquote off the shelf so that they can be made in advance, frozen, shipped to wherever the patients need them, given to the patient there, and can be, just as effective. So just like, I say for the statin, the pharmacy doesn't make this for every patient. We want to do the same thing for cell therapies. And the way that my group has been doing this really for the last 20 years has been to use these human pluripotent stem cells. So this Kat introduced, we started this work with what are called human embryonic stem cells. In the last 10 to 15 years, this has moved into what are called induced pluripotent stem cells. These are cells that can make all the cells and tissues in the body, okay? I'm a hematologist. I'm interested in making blood cells. We spent a lot of time trying to make hematopoietic stem cells, very challenging. What we've been very successful at is making this other population of immune cells called natural killer cells, or NK cells. We can make these quite routinely, and we can scale them up to clinical trials. And I'll show you a number of clinical trials has been done for this. They're different than those T cell therapies in that, well, T cells have to be primarily matched to a specific patient's. NK cells don't. They have a different receptor system, so my NK cells will kill your tumor cells and vice versa. So if we make these in the lab, they don't have to be matched to a specific patient. These are so-called allogeneic, again, quote unquote, off-the-shelf treatments. And so this is kind of the process we go through in the lab. We can grow these undifferentiated cells. We can engineer them to make better NK cells. So when I first came here, I talked about making super NK cells, and I did that for a talk. I still remember Scott Lippman, who was our former head of the cancer center, would keep asking me, so when are you going to have those super NK cells? Well, we got them, and I'll show you some of those results. And we can make these into large numbers of engineered NK cells. And so I hope you don't mind. I'm going to show you some pictures of mice with cancer. But this is what we do in the lab. Okay, so we use a special type of mice. These are immunodeficient mice that we can put human tumor cells and human immune cells in them. The tumor cells are essentially bioluminescent. They glow. So if you see that second row, the tumors glow and they grow and the mice die. Okay, the middle rows, they get sort of ordinary NK cells from different sources, not engineered. And they do a bit better, but they still progress. But we developed this particular type of receptor, another type of CAR that recognizes this as an ovarian cancer model. Those mice do better, and the green line on the right shows that they live longer. And I'll show you that this has actually moved into clinical trials. We've done this with another type of cancer. This is a leukemia model where, again, we can just use regular NK cells and the mice die. If we use our engineered NK cells, the tumors go away. And in this case, we actually cured 3 out of 5 of the mice. So this is work we've done over the last decade or so in my group. We previously collaborated with a company here in town called Fate Therapeutics. And this was actually, as Kat mentioned, the first iPS-derived NK cells were given here at UCSD back in 2019, so we're over 6 years ago. We have permission from the patient to use this. This is me with Sandeep Patel, who is the PI of the trial. I had a conflict of interest, so I couldn't actually be part of the trial because this was based on our work, and there was licensed IP from this. But as I have here, that's actually the first dose of cells, and that was about 20 years of work into that bag. So that was about 6 years ago. More recently, so Fate just published another trial which uses some of these now super NK cells. Those first ones were non-engineered. They just had to get them into the clinic that way at first. These now have these improvements based on some of the technologies we've developed in our lab. This is now, again, for a certain type of lymphoma. 86 patients now treated on this trial. 20 of them had actually failed the previous CAR T cell therapy that we talked about. And over half of these patients had remissions or responses based on this, some of them for a year or more. This was just very recently published, and hopefully more of that work coming up. So then we want to do, as Kat mentioned, again, we want to do more of this work here. And what Sanford Stem Cell Institute has really done is help to support our Advanced Therapy Center. Of this, we have what you mentioned is the Advanced Cell Therapy Lab. This is what we talk about as a GMP facility. So that's where we can actually make cells that are suitable for clinical trials. They won't let me take the cells that we give to mice to give them to people. They have to be made under special conditions. As you can see here, sort of like the stuff you see on TV with people in bunny suits and very clean, controlled environment. And so what we're able to do with this ACTL and the Sanford Advanced Therapy Center is to develop more of these novel cell and gene therapies. So we work with both biotechs and academic groups. We can do the work either to get cells into clinical trials or to actually do it for clinical manufacturing. Right now, we're limited to just doing cell therapy. We want to develop space, just was talking a little bit earlier today, to actually do the gene therapy vector production as well. And then again, we have through the Sanford Stem Cell Institute, the Alpha Clinic, and all the pieces enabled to move these from the lab to the clinic. So this is just an example. This wasn't actually a cell product that we made here, but we helped maintain these cells and get them to the clinical trial. I think we'll hear more about this later of a company called Neurona, where they're actually doing a cell therapy for epilepsy. Not my area, really interesting, really amazing results. So, these are the type of trials we want to be doing more of here. As another area of support, I just want to mention, if you've all heard of CIRM, California Institute for Regenerative Medicine, they now have what they call their Cell and Gene Therapy Biomanufacturing Network, which we're part of along with 8 other academic groups in California that provide some support to build up this type of infrastructure, as well as workforce. We really need people to actually do the work. Tiffani may talk more about that as well. But, getting people interested and trained in this area is really important. These are the people in my lab that do all the work, and I'll just stop there, and I guess we get questions later. So thank you.
Thank you so much. All right. So you see this incredible environment that we live in here. Thank you so much, Dan, for summing up more than 20 years of work and doing it extremely efficiently. So next we're going to chat with Karen. So Karen Christman co-leads the Sanford Advanced Therapy Center and is also a powerhouse. She's in bioengineering and was recently named to the National Academy of Inventors. So you heard how inventive Dan's work is. Well, we've got Karen Christman, who's equally innovative and inventive, and we'll talk about her discovery of how stem cells need a happy home. They need to be supported by something. Well, I remember when Karen started to talk about this extracellular matrix, and we said, well, how good is it really, though? And it turns out it's good for a lot of stem cells to stabilize them and to keep them in the places they need to be, particularly in cardiovascular disease. So we heard from Dan on how important it is to use regenerative medicine-based therapies for cancer. And so that's the number one cause of death if you get to be over 85. But if you're 85 or under, it's actually cardiovascular disease. So Karen Christman, who is our co-director of the Sanford Advanced Therapy Center, will tell you everything that she's done really as a formidable force in the bioengineering field to change what happens after heart attacks. When people have heart attacks, sometimes the oxygen doesn't get to that part of the heart. It doesn't get properly perfused, or the blood flow doesn't get in there. Karen's got a solution. When people have problems with peripheral vascular disease, their arteries get clogged, Karen has a solution. So we always look to Karen for solutions, just like we look to Dan for that. So they were the dynamic duo. And then we'll move on to our friend at Thermo Fisher to talk about what's unique about this place. If you think about going to Napa, do you remember when we went up there? They make the best wine because they have the people that make all the stuff to gather the grapes. It isn't just that they have the special winemakers. They actually have all the equipment, all the reagents. We live in a special place here, and we're really lucky to have such great colleagues. So Karen Christman will tell you about what she's doing on the ECM front. Thank you.
Okay, thank you, Kat, for the introduction. And yeah, good evening, everyone. So it's my pleasure to tell you about some of the work we've been doing in my lab, and then also now more recently part of the Sanford Advanced Therapy Center on designing and translating injectable biomaterials for regenerative medicine. So just required disclosures, I'm co-founder of a couple companies. But as Kat mentioned, one of the big focuses of my lab is on heart attacks or myocardial infarction. And I'm guessing that nearly all of you either have a family member or know someone who has had a heart attack. And so it's a huge problem in this country as well as worldwide. And as Kat mentioned, it causes a loss of blood supply caused by a blocked artery. And then that portion of the heart essentially dies. The cells die off, and it's a very inflammatory environment. You get a lot of inflammation, and then that leads eventually to scar tissue formation. So just like I'm sure everyone has a scar somewhere from when they were a kid, hurt themselves, that same process happens inside the heart where you get this scar tissue formation in the region of the heart attack. And the heart basically undergoes this negative left ventricular remodeling process where the heart gets larger and then can't pump blood effectively, leading to heart failure. So there are really, unfortunately, no good solutions for this. And if you're an end-stage heart failure, you're left with a heart transplant or a left ventricular assist device or mechanical pump. So people have turned to regenerative medicine strategies for the heart. And what was initially tried a couple decades ago was cell therapy. And there was a big push about 10 years ago to translate a lot of different cell therapies into the heart. But unlike some other indications where cell therapies have done really well, and the heart, they didn't actually do that well. And so a lot of what we think the reason for that is that they're not provided a great home. So if you think about where these cells are injected into, they're injected into the infarct or the border zone around that heart attack region. And this is what it looks like under a micrograph or a microscope. So it's a very dense collagen scar, just like that scar that you might have on your arm from when you were a kid. And so this provides basically incorrect cues to the stem cells that you're injecting. So it provides incorrect biochemical cues because the natural scaffolding or the extracellular matrix that the cells in the heart sit in is not just collagen. It's a lot of different proteins and even sugars, polysaccharides. And so you're providing abnormal biochemical cues, but also abnormal physical cues. So the scar is much stiffer than the normal extracellular matrix or normal scaffolding. And so I think it's not too surprising that you inject these cells. There's also low blood supply, and so they don't do very well. So we take kind of an opposite approach to regenerative medicine. Instead of trying to replace the cells that have been lost, we try to replace the extracellular matrix that has been degraded and lost also after a heart attack, with the idea of providing a more healthy microenvironment that the body's own cells, stem cells, blood vessels can come in, repair that region, leading to more heart muscle and less of that scar tissue. So we think about trying to recreate this new microenvironment and to try to provide a new template for healing. And so we try to do that with injectable biomaterials. So this idea of using biomaterials for regeneration is not necessarily new, but people haven't typically done this really with injectable forms. And they haven't really done with this with the heart. But there are other examples. For example, there's peripheral nerve grafts that are just biomaterial scaffolds. There's also a vascular graft, a tissue injury vascular graft that's just, again, extracellular matrix that actually was just FDA approved last year. And so what in general this does is if you design a material appropriately, it can promote the body's immune system to promote a pro-regenerative as opposed to a pro-inflammatory response. And that stimulates endogenous repair, stem cells to come in and heal the tissue. And one of the advantages of these approaches is that in general, it can be much more cost-effective than a cell therapy. So Dan mentioned CAR-T, which is $500,000. The lowest end I've heard for the heart is about $10,000 or tens of thousands of dollars. To give you an idea, the material that I'm going to show you that we invented for the heart, it's about $500 for one vial to treat a patient. So orders of magnitude lower cost for patients. And it's also not a living product. So cells, while they can be great for many indications, do have issues with that it's a living product. You have to worry about storing it, shipping it, preparing it. Did the cells change? Here, you can have a material that's off the shelf. It's not going to change upon storage conditions and ready to go. So one of the first technologies we developed to treat the heart in this idea of trying to recreate the natural extracellular matrix that's lost is what we call our myocardial matrix hydrogel. So I'm going to show a video in a second here to give you an idea of some better visuals of the process. But in short, we essentially take what nature has given us, which is pig hearts, which are largely a byproduct in this country. And if you isolate just the extracellular matrix that you see in the the second image there. So we stir it around in a detergent, actually a detergent that's found in some cheap shampoos, that you can strip out all the cells, leaving behind kind of that ghost white structure there, which is the extracellular matrix of the heart. And then we further process it to get into an injectable form that when we inject back into tissue, it reassembles back into a porous and fibrous scaffold, very much like the original extracellular matrix. If you look at it with a scanning electron microscope, it has this nanoscale fibrous architecture, very similar to the original cardiac extracellular matrix. And we designed this material to go through a catheter where you're just accessing at the femoral artery in the groin, snaking it up to the heart, and then you can perform minimally invasive injections without general anesthesia, without surgery, just using a mild sedation in a cath lab. So next I'm going to show you, again, just like I said, a visual, a video that was the person talking is one of my former MD PhD students who's actually now a cardiologist at Vanderbilt with her own lab.
Hi, my name is Jean Wang, and I'm a first-year MD PhD student in the Christman lab, and this is how we make our therapy for treating heart attacks. So we get heart tissue, and the first step is we chop it up into tiny pieces, and we put it into a bath, like a beaker right here, and stir it up with detergent to remove all the cellular contents. After a couple days, we rinse it out to remove all the detergents, and all we really have left are the structural proteins that make up a tissue. So we take that and we freeze dry it into this styrofoam-like substance. It's really light and crispy, so it's freeze dried, and then we take this and we mill it into a fine powder that looks like this. We take that and digest it with an enzyme to liquefy it. You could see that there's liquid at the bottom of this, and this little bead is just stirring it up and keeping everything well mixed. And then finally, we take the liquid form of this and we inject it into the heart, into damaged heart tissue. And once the liquid hits body temperature, it forms a gel that looks like this, and it no longer flows anymore.
And so after I'm done too, you can, in the back, Maria, one of my current PhD students, actually has some examples of the hydrogel just in a vial if you'd like to take a look at it, as well as some images from our lab. But we, over not quite 20 years, as Dan, I'd say about 15 years of work, we have really extensively studied this material in the, in vitro, so in the lab, small animals, large animals, and then that's culminated in a phase 1 clinical trial that was also completed right before the pandemic. So this is the image there, it's called Ventrogel commercially. It comes just like that, a lyophilized cake, freeze-dried cake. You can add sterile water right when you're ready to use it in the cath lab, rehydrate it, and then you can deliver via the catheter that I mentioned. And so this trial had 15 patients, initially just a safety and feasibility study. So patients were between 60 days and 3 years after their heart attack. It was delivered, like I said, this minimally invasive catheter approach, and then they were followed out between 3 and 6 months. So all patients were treated, so looking at compared to baseline, but overall good safety results, which was great from, obviously from a safety perspective, but also this was the first extracellular matrix hydrogel version. There have been patches of extracellular matrix, but not a hydrogel injectable version. And this was the first that had gone into patients, and obviously the heart is a pretty high-risk organ, so showing safety, I think, really opens up the potential for using this not only in the heart, but I'll show you a couple other examples and other indication as well. But of course, we looked at hints of efficacy and showed at 6 months we got significant increases in exercise capacity, decreases in heart failure symptoms, and also 80% of patients we evaluated using MRI, 80% of patients we were able to basically either prevent the heart from expanding larger and going into heart failure, or even had a reduction. So good initial phase 1 results. Obviously needs to be evaluated in larger randomized controlled trials. Unfortunately, J&J decided to stop making the catheter that we're using, but there are some new companies coming online in about, I'd say, the next year. But in the meantime, while we're waiting for that catheter, we have 2 new phase 1 clinical trials that are going to start enrolling, hopefully by the end of this year. We're shooting for in fall, that use a surgical-based approach when patients are already undergoing a surgery and we don't need the catheter. So one is in CABG or bypass patients that can't be fully revascularized, meaning they need to bypass 3 blood vessels in the heart. Maybe they can only bypass 2, and those patients are known to have worse outcomes. So we're going to inject into the region of the heart that cannot be bypassed with a vessel, and we'll inject the hydrogel in to stimulate the body's own cells to come in to stimulate new blood vessel growth and hopefully improve outcomes in those patients. And this is partially funded by the UC San Diego Stem Cell Institute, and we're currently also looking at other funding sources to complete the trial. Other trial that'll start later this year is in congenital heart defect patients, so those with hypoplastic left heart syndrome. So this is where essentially babies are born with only the right side, right ventricle of their heart, whereas the right ventricle is supposed to pump blood to the lungs. The left ventricle is supposed to pump blood to the whole body. So what happens is they undergo a staged operations to essentially refigure the anatomy so that the right side of the heart does all the work to the lungs, but also to the systemic or to the body. Systemic circulation. And so this actually, as opposed to these kids dying as babies, that allows them to start to grow up. However, the right ventricle does fail because it's not meant to do this. And so we have nice preclinical data showing we can improve right ventricular function. And so the goal is to inject into these babies to prolong their cardiac function so that they can grow up and get an adult heart transplant, because it's much more difficult to get a pediatric heart transplant. So the CABG trial is in collaboration with Tony DeMaria, who's a cardiologist here at UCSD, as well as Jay Travers, who's at Minneapolis Heart Institute. And then my collaborator, Mike Davis, is at Emory for the congenital heart defect trial. So that's the work we have ongoing and translating to the heart. I did want to mention just some of the other applications. So we started with the heart and got us into these extracellular matrix hydrogels. And since we had such great results with that, it really expanded our options into treating other tissues. So one that Kat mentioned, peripheral artery disease, also caused by atherosclerosis and narrowing of the blood vessels, typically leading to issues with the legs. And this causes muscle atrophy, degeneration, and low blood flow, causing things like ulcers and gangrene and eventually amputation. So we have great data with it now, a skeletal muscle version of our hydrogel, and since we're treating here ischemic skeletal muscle or skeletal muscle with low blood flow. And in collaboration with Shami Mahmood, we're currently working on an IND, so what you asked the FDA to start a clinical trial. We've already successfully completed the pre-IND, which is kind of the stage before that. With Dr. Andrew Vahabzadeh Ha here at UC San Diego, who is an ENT surgeon. We've also been looking at using this material to regenerate skeletal muscle in the tongue for patients that have head neck cancer, get irradiated, and then they get a scar in their tongue and it makes it difficult to swallow or talk. And so we've shown in preclinical data that we can get regeneration in less scar tissue. There's actually an image in the back of the room you can see later. And so we're also working on a pre-IND for that application. And then about a third of my lab actually works now on women's health. About 8 years ago, Dr. Mariana Alperin, who's also in the Sanford Consortium for Regenerative Medicine, who's a urogynecologist, approached me and really opened my eyes to kind of the areas of women's health that are really underserved and really need new therapies. So together we've been looking at extracellular matrix hydrogels to treat pelvic floor disorders, so to regenerate the pelvic floor muscles that get injured, particularly after birth, can cause pelvic organ prolapse and urinary and fecal incontinence. And we've also have some recent data on extracellular matrix materials to treat symptoms of genitourinary syndrome of menopause. And so I think there's a lot of exciting applications. We're hoping to translate these technologies also into patients in these new indications. So just for the last minute, I wanted to just mention, how we really translate these therapies. And I think it really comes down to people and partnerships. So here's the current people in my lab. I have had been very fortunate to have outstanding both graduate students, undergrads. We have a lot of undergrads too in the lab, as well as postdocs who are very interested in translation as well. And as you can see, we work very closely with many clinical collaborators that are really instrumental in working with us as engineers to translate therapies into patients. And then we've been very creative with our partnerships across the stages that you need to get to patients, from early stage research, like small animals, to translational research, large animals, and safety studies that are needed to go to the FDA, to clinical grade manufacturing, like GMP manufacturing that Dan mentioned, to phase 1 trials. And I think, this really you need a partnership. Initial studies have been federally funded, but we've also had some fellowship funding. CIRM has nice funding that can fund large animal translational research and manufacturing. And then I think ultimately you do need to partner with companies to go into trials. But what's really great about the Sanford Stem Cell Institute is that it bridges all of these things. And I think particularly with the current federal funding, concerns, that it would be even more important to have these creative partnerships. And with the Advanced Therapy Center, I think we really sit in that middle for translational research and clinical grade manufacturing, and hopefully get more things into patients. So with that, thank you very much, and look forward to the discussion.
All righty. So last but not least, we'll just have Tiffani Manolis come up and give just 3 minutes, to tell us about why we can do these things with Dan and Karen and our team. We live in the land of Thermo Fisher. Thermo makes all the reagents for science. Tiffani actually is the vice president of cell biology for Thermo Fisher. She has 25 years of experience in the industry to make us look good. Thank you, Tiffani. Before we go on to Tiffani, I just wanted Matteo and Christian and Marikar and all members of our Sanford Stem Cell Clinical Center to stand, Sheldon, to see when these brilliant people, Dan and Karen and others, make these new therapies. This is where it goes. Do you want to stand? Please. I know, I'm sure I missed people. And then we've also got Michelle Ordegaro, who's already standing at the back, who's our executive director for the Sanford Stem Cell Institute. And then our whole team, so we've got Danielle and Bobby and Catriona, Nick, Kirsten. We've got a whole team behind you. So we'll be taking any questions at the end. Thanks. Thank you for being here, Tiffani. Just 3 minutes.
Just 3 minutes. So let me start by saying thank you for the invitation. At Thermo Fisher, we think a lot about the problems and the challenges that face patients. And we frequently think about all of the challenges that patients face in a given day, and the number one challenge being your health. So you may have 1,000 challenges in your day, but when your health becomes the challenge, you really only have one challenge in your life. And so one of the things that we think about at Thermo Fisher, it was so well described in the previous presenter's slide, is how do we take early stage discovery in cell and gene therapies and bring them all the way to the patient and really democratize cell and gene therapies. You heard from Dan, sometimes it's $500,000 for a cell and gene therapy. It's a fantastic opportunity for us to collaborate with researchers and small biotech and large biotech to really drive advances in science, to bring the costs of these therapies down and increase the number of patients that can get access to these therapies. And in many cases, I felt for the mice. 3 out of 5 of those mice lived. We're looking for that and even better in patient outcomes as we work. So a little bit about Thermo Fisher and maybe our position here. I am the vice president and general manager of the cell biology business. Our headquarters is just up the road on a beach called Carlsbad. Fantastic place to be. Just about 6 weeks ago, we opened a huge collaboration center for cell and gene therapy customers. And we have a tremendous amount of resources there from our scientists and our program managers, our regulatory, our quality teams, and supply chain to really help our customers go from the bench to the patient's arm. And I invite you to join us there anytime. Some of the team is here, so you can reach out to us in the cocktail hour and come on up to Carlsbad and see what we have to offer. But it's there that we work side by side with customers to really revolutionize our products to make sure that we can get more and more patients into trials and more patients their therapies. So on behalf of Thermo Fisher, thank you so much for the invitation. We're really excited to continue the strategic partnership with UCSD.
All right. Well, thank you so much, Tiffani. That was amazing. Yes, we do want to work with you a lot more, and there are great restaurants up in Carlsbad, so we might have to just avail ourselves of Campfire or Choni. So for the last but not least person that we'd like to speak is somebody that had the most complicated clinical trial. His name is Justin, and this is why we do what we do. When we were going to start the study, Christian and Matteo and everybody on the team said, of course we can do this. Why wouldn't we, even though it's the most complicated? Because we have to, we must. So when I was a bone marrow transplant fellow at Stanford, I was the person who looked for neurotoxicity of a drug given quite commonly to suppress the immune system after cellular therapy. I was used to doing this for bone marrow transplants, not so much for the first stem cell derived therapy for stroke. And this is with Gary Steinberg. I was actually on that paper in the Journal of Neurosurgery. And it was an unusual position to be in, but I realized how important it is to use regenerative medicines beyond cancer into devastating diseases, be it heart attack, stroke. But something it never occurred to me, epilepsy. Epilepsy, intractable epilepsy, actually really affects people's function. And not having the ability to drive is really debilitating, or to travel across the country. Well, the person you'll be hearing from was on the Neurona clinical trial, and he's quite public about why stem cell therapies, and particularly regenerative medicine from pluripotent stem cells, really matters. I think we have Justin here. Hey, Justin, how are you?
Doing all right. How are you guys?
Thank you so much for joining us. I know you met our friends in the space industry at Redwire, so I was quite astonished to hear that you've been doing your own road trips.
Yeah. Yeah, that was a fun one to go check out and just see how they're actually doing stuff, going up to space and doing the different testing with the cells.
Now, you started your life at Louisville, Kentucky, is that right?
Right across the river, Floyd Snow.
Right across the river, where there are more horses than people, right?
Yep.
So, Justin, you moved here to California, where you've had these unique opportunities to get a cellular therapy. Can you tell us a little bit about the process, what you experienced, what you were thinking about when you went into the clinical trial, and how things have been since? You've got your whole team here cheering for you, but how have things been?
Well, things are definitely great. Right from the start, I didn't really think much about it, I guess. I got asked if I was interested, and it's just like, of course, sounds great. And it wasn't until after a while I finally really thought about how big of a surgery that would actually be. But the first thought was how much of an advancement this would be in the medical field, and just being able to help so many other people with this, not just my condition, but it just seems like there's such a wide branch that all these can go out to. So I started off having seizures in 2007. I had just become a scuba diving instructor, so I had to give up that career dream. I'd also been a competitive swimmer most of my life, so I wasn't able to do that anymore. So it had a pretty big effect on me, not being able to drive, of course. And then after having this operation, my life's almost completely back to normal. Thank you. Yeah, it's almost completely back to normal. There's, of course, a few things I've got to be a little more careful about, but being able to manage trips on my own, just not being so worried that if something does happen and I'm at home by myself, or just possibly out somewhere that's a little further away from people. I love photography, so I tend to get out in nature a bit, so helped a lot with that as well. But yeah, it's been an incredible experience.
So when I was at Stanford and was involved in that neurosurgical clinical trial, we couldn't make the cells into what we wanted to make them as much. Now you've got a really engineered cellular product that came from pluripotent stem cells and was made into a GABAergic product to suppress the seizures. Before you got that cellular therapy implanted into your brain by our clever neurosurgeons, and specifically we have a brain tumor and neurorestoration program led by Alex Kalesi. How many seizures were you having a day?
Only about one. It was usually somewhere around 30 a month. Sometimes there were days I didn't have any. Sometimes there were days I'd have a couple. But usually about 30 a month. And now it's down to maybe 1 or 2 a month.
Okay. So now you're able to drive and travel, is that right?
Yes.
Yes. And you mentioned something, Justin, that I've noticed with you before, because you spoke at our Sanford Stem Cell Institute and you were here to impress NASA with everything you've done, and they were blown away, by the way. They want to invest more in human research and helping humanity get better therapies. You mentioned one thing is you want to help other people by being involved in clinical trials. Were you always that altruistic? Is this a unique thing that came up because of this opportunity, or what happened?
No, it's actually kind of a family history, I guess. I love it. My dad's a father. I've always had a huge interest in medicine. It was when I was 16, I found out my grandfather was actually the one who invented the time release capsule. And so I keep learning about these different things. So it's just really been something that's just, I guess, ingrained into my family.
That's fantastic. Well, Justin, we really appreciate you being here by Zoom, especially considering that you can't be here because you're traveling, which you couldn't do before.
I was almost going to be out there this week, but
yeah, well,
decided not to hang out with my parents a little more.
So I wanted to show you something about Justin here, and this is something that Lee Stein and I have talked about a lot. AI does not supplant natural intelligence, and it won't be for doing that for a long time because Justin's intelligence is regenerative, and we've enhanced that regenerative intelligence with new stem cells that function a little bit better, but also there's no altruism in AI. We learn from each other. A is for altruism, and that's what trumps everything. So thank you, Justin, for leading the way.
Of course.
As our star of the hour, we'll have you stay on there, but we'll also invite our 3 speakers up here so they can be grilled, not by me, but by everybody in the audience, because I tend to go on ad nauseum. And then we'll be here for the next 3 hours. I know that a lot of people have questions, and they may have questions for Justin or Karen or anybody up here. Does anybody have any questions that you'd want to come to a mic for, or Kirsten can bring a mic to you?
I'm just curious on the vascular application of this. How much of a component is there, like a temporal component of this? In other words, before the fibroblasts really set in, how much of like an immunosuppressive opportunity is there to kind of apply some immunosuppression before the extracellular matrix kicks in? And is there an opportunity to kind of apply some of this stuff on a temporal fashion before all the scar sets in?
Yeah, so great question. So we've looked very much at timing. I'd say that first trial in heart attack patients involved kind of subacute to chronic. We've also looked in the lab where we can study in more detail on subacute and chronic stages. And it can work in both stages. One, it's more interfering with scar tissue deposition, whereas in the chronic stage, we actually can get remodeling of the scar tissue. And then actually, if you want to talk to Maria, who's going to be in the back sheet, we have a new version that I didn't mention today that you can deliver into the bloodstream. And that she's working on it for multi-organ dysfunction syndrome, but we've also looked at it for acute heart attack, meaning deliver it immediately, like somebody comes in, gets a stent, or angioplasty stent, deliver it then to try to basically salvage the myocardium and to try to inhibit some of those inflammatory stages. So there's slightly different mechanism of action depending on what time, but we've seen you can impact, different stages.
Such a great point, though, because there is a huge inflammatory infiltrate that promotes the scar tissue. And so when I used to do bone marrow transplant, there was a lot of work on trying to bring in cellular therapies that would suppress that avert immune response with mesenchymal stromal cells. It just didn't work very well because they didn't know what to do when they got there. And that's why Karen's work is so fundamentally groundbreaking and game-changing. Any other questions? Yes, we have another question at the back.
Hi, thank you. My question is with regard to regenerative cell therapy. Do you know if there are any efforts for multiple sclerosis with myelin regeneration?
Yeah, so there's a lot of interest in ongoing clinical trials for autoimmune disease in general. And I believe there are trials for MS. Using those same, actually, the CAR T cells that I explained that were complicated and expensive, but they do a good job of killing B cells. And so most of the trials have actually been done for autoimmune disease, systemic lupus, but I think there are some for MS. And those CAR T cell trials are being done for MS and other autoimmune disease. And I know for the other diseases, the responses have been remarkable. I mean, essentially all patients have had complete remissions. There's been some other types of cell therapy, and I know we've done some trials here. I can't actually remember the product for MS as well, and I'm sure we can get to that info, but it's definitely an area where there's ongoing work.
Yeah, so Jennifer Graves has trials for MS, and we're working very closely with her through the Alpha Clinic. This is the CERN-funded infrastructure that allows us to go from the bench to bedside and get these new therapies made. So we're working really closely with Jennifer Graves. There's a CD19 targeting CAR T cell trial from Genentech and Poseida that we're trying to get in the door. CD19 is expressed by B cells that have infiltrated the brain and are contributing to those lesions in multiple sclerosis that make it progressive. So she's working really hard with our team. Dan runs the Cell and Regenerative Medicine Service, so we'll be seeing those patients as part of our service responsibilities. So we're unique in that we have a Cell and Regenerative Medicine Service, so we can look after people when they have complications from CAR T cells. But there's a lot still to come on the cellular therapy front for autoimmune diseases. And as Dan was mentioning, this is a huge new target. We used to think everything had to be about cancer. Well, it turns out autoimmune disease is pretty tough to deal with, particularly MS. So there are a lot of opportunities there. There are antiviral strategies targeting MS because it looks like Epstein-Barr virus that a lot of us get, but some people have a massive immune response against, maybe a target. So that was the other therapy that we'd been doing targeting MS, is targeting that virus or the avert inflammation caused by that virus. So you'll see a lot more next year when we repeat this. Any other questions? Yes.
Great talks and a tremendous story, Justin. Just a quick question. So we have seen Dan and Karen mention a few industries, Fate, and then Ventrix Bio, and then obviously we have Thermo Fisher. Besides getting expedite the entire clinical trial process of getting therapies to patients faster and cheaper, what are actually some of these things that you're looking for in a successful partnership with the industry from both and also Justin's perspective?
Well, let's start with Justin because timing is everything, especially when you're dealing with a seizure disorder where you have 30 seizures per month. So Justin, how would you think we can improve on the process when it comes to partnership with companies through the Sanford Stem Cell Institute and companies like Thermo Fisher?
One of the biggest parts, I guess, is just bringing the awareness to it and that it's a possible option to go with, and how much of a better option it could possibly be. Seems like it already is, but just want some more money has been put into it for the actual studies, making sure that it is going to be completely safe for different people and what all they can use it for. So it's just a huge step forward that I hope we do keep putting all of the money and time into, and can't thank you guys enough for doing that.
Thank you, Justin. So we're going to bring this to Tiffani from a thermo standpoint. I mean, these are tough therapies to make. What has fueled your zeal about doing this, almost on steroids? Because thermo is really leading the charge here for people like Justin.
Well, I think the patient is really the primary fuel, and many of us have spoken about heart attacks and that affecting patient populations and ourselves. We're patients. Cancer is very similar. Autoimmune is a really exciting space where cell therapy is going. But when we think about partnership, we are trying to work quickly. So speed is definitely a piece of partnership. Safety is a piece of partnership. So making sure that we can support customers from a regulatory perspective as these drugs go from bench to patients' arms very quickly. That's a process, and we want to be sure that that drug is very, very safe. Those are a couple of the ones. Outcomes and impact is another one, and that's really back to the patients. So when we think about partnership, those are the things that are kind of top of mind for us at Thermo Fisher.
So one thing, as the Institute director that occurs to me is we have industry, thank you, academia, and people who also start their own companies here at UC San Diego, but also philanthropy. Usually to get anything into clinical trials and then all the way through to FDA approval, we have to have industry funding, academic funding, and philanthropic funding. And that tripartite stool, the three-legged stool, is how we get things to FDA approval. So all of us can make a difference. We can invest in our own futures and make this the healthiest place on the planet. So I'm just wondering for last words. Karen, any last thoughts?
I completely agree about the, three-legged stool I didn't mention, but like our congenital heart trial is funded philanthropically.
It's amazing.
And so I think having that partnership is really important, particularly because the really large companies, at least it depends what area you're in, but say in cardiovascular, they're not interested until like phase 2, late stage trials. So you really need, I think, kind of that being creative and getting funding sources from these different things to get through that phase 1 trial.
And then Dan?
Yeah, I mean, I just want to bring up that, with all of this that we're talking about and doing this here in San Diego, I mean, as was mentioned, I came to San Diego 9 years ago. Yeah, it's a great place and you have the ocean here, but it's really the scientific community here, right? With not only UCSD, Salk Institute, Sanford Burnham Prebys, Scripps, and so on, and all the biotech and Thermo Fisher and everybody else here in a really accessible area. I mean, we see each other. It's not, I think, like, Boston or the Bay Area where it's very dispersed. I mean, we all kind of know what's going on. But even in the last decade, I mean, the amount of biotech that's grown here has just been phenomenal. And I think there's still so much more we can do now with the Sanford Stem Cell Institute and UCSD. Of course, we want these trials to be done here, but it's really all the pieces are in place. And one of the things that makes this unique is, UCSD is the only academic medical center in one of the largest, populations in the country. And it's very unique for this to be the only academic center. If you go to LA, New York, Boston, whatever, you have a half a dozen of these. And so there's, we've done a lot already, but, I think there's really so much more that we can be doing here.
Great. Thank you so much, Dan, Tiffani, Karen. Justin, final word. Are you going to start swimming again?
Yes, I actually had my nephews over here today, so played in the pool with them and hopefully getting back to actually doing some real exercise with it.
Fantastic. Well, we're so glad you've got some new strokes going and that you're doing so well. I think we'll finish it here. Thank you, everyone, for being here for really a fantastic Mind event. Thanks for a great event.